For the next presentation, we're moving to Sudeep Popat, who is from the United States at Clemson University. Dr. Popat is an assistant professor in environmental engineering and earth sciences at Clemson University. His research group focuses on anaerobic biotechnologies and electrochemical technologies for resource recovery from domestic and industrial wastewater. His group is currently working on a project funded by NASA to develop microbial and electrochemical technologies for resource recovery from wastewater during space missions. And his presentation is engineering the electrochemical ammonia stripping process for nitrogen recovery from source separated urine.
Right. Can you all hear me okay?
Yes, very well.
Right. Thanks, Abe, for the introduction. So I'm going to talk about the work that we've been doing in the area of electrochemical ammonia stripping for nitrogen recovery from source separated urine. I want to start out first by acknowledging my PhD student, Phil, who did most of the work that I'm going to talk here about. But this idea about electrochemical ammonia stripping in my lab, Aoshi was the student who first started exploring this for animal processing wastewater, and we have since translated some of our work to source separated urine as well. A lot of the work that we are doing on urine processing is funded by NASA EPSCoR program and the South Carolina Space Grant Consortium. And before I move on with the talk, there's one more acknowledgement that I would like to give, which is to Nancy Love. So this work that we started doing on urine sort of first started occurring in my mind when I toured their facility at the University of Michigan for the AESP conference. And then later on, she visited here at Clemson last year for a seminar, and she mentioned to me about the Research Summit. And I couldn't go last year, but was really looking forward to joining for this year. Unfortunately, it has to be virtual, but I look forward to being part of this community for many years to come. So with that, I want to introduce what electrochemical ammonia stripping is. It is basically a process which involves the use of electrodes to alter electrolyte pHs, right? And so in this case, you can have reactions that you can do on the anode and the cathode that will acidify the anolyte and basify, or increase alkalinity in the cathode chamber. And so we are relying on reactions that change pH. And this is of importance because then you can do things such as strip ammonia out of solution by not adding chemicals, but just rather relying on electrodes to produce your base. In this case, what happens is that if you try and feed urine through the anode chamber of an electrochemical cell where you're doing water electrolysis, ammonium will move through a cation exchange membrane to the cathode, where really from reduction of water, you're producing hydrogen gas, but along with that, you're also producing hydroxide ions. So this is a chemical-free stripping of ammonia that you realize here by selectively transporting ammonium through a membrane from the urine and then converting it to ammonia gas, which can then, depending on how well you are able to control the pH on the cathode, you can have the equilibrium shift towards ammonia gas, and then you can have multiple different ways in which you can recover that ammonia gas. You can have stripping columns that are adjoining the electrochemical setup, or you can do other methods of recovering that ammonia, such as transmembrane chemisorption, and more recently, the use of gas permeable membranes has been shown by William Torpey's group as well. One of the questions that I had always had, and this is again, explored somewhat in the literature already, is that when you try to do electrochemical ammonia stripping from urine in this way, the presence of chloride ions in urine will cause an issue. And the issue is that in addition to oxidizing water to produce oxygen gas on the anode electrode, you will also, in all likeliness, on most electrodes, produce chlorine gas from the chloride ions that are present as well. And this could be a good thing or a bad thing depending on what you're trying to do. One of the things we know for sure is that would most likely achieve some disinfection of the urine, but on the downside, it will also lead to formation of maybe some disinfection byproducts. The reason why it's important is that the theoretical potentials of both of these reactions that will occur on the anode are fairly close. And so that's why most electrodes, you have to, there's work that needs to be done on electrode selection if you want to avoid this. But one of our ideas was, can we look at, can we engineer the process in a way? Can we look at the process configuration and figure out ways in which we might be able to avoid that reaction? I'm showing here data from a study from Will Torpey's group that clearly show that in synthetic urine, you can actually measure detectable chlorine. You don't do that so much with real urine, most likely because you are forming a lot of these disinfection byproducts, maybe that you don't pick up. And so what I want to mention is that, we have a hypothesis of how we think we might be able to engineer the process to not actually have urine production. And it's actually a fairly simple modification. We know that the pH increase happens at the cathode. And in the anode fed operation, we are relying on transport of ammonium to the cathode to be able to convert that to ammonia gas that can be recovered. Instead, if you feed the urine straight into the cathode and still have a cation exchange membrane that separates the anode and the cathode, we avoid the occurrence of chlorine at the anode chamber. And by doing that, what we're doing is we are preventing the production of chlorine gas. That's our hypothesis. In this case, you still have to maintain electron neutrality. And so here you sort of end up relying a lot on the salt solution that you feed through that counter electrode. So typically we would feed something like sodium sulfate or maybe some other sodium salt, which would at least in experimental case, we'll be able to do a proof of concept with, where we would then rely on transport of sodium from the anode to the cathode to maintain electron neutrality. Remember that when you're doing these reactions, when you're moving electrons from the anode to the cathode, you have to have electron neutrality that is maintained. So our hypothesis is that this configuration will reduce chlorine production significantly. And so what we've done is we've looked at how anode fed and cathode fed electrochemical cells for electrochemical ammonia stripping perform in terms of these important metrics. How much ammonia can be removed and recovered, how much chlorine is produced, and how we might further engineer the system. So this is a photograph of our system. It's what I would call a two-chambered electrochemical cell. This here is showing anode-fed operation. So we have synthetic urine fed through the anode chamber. We have a carbon cloth anode that we place in this. So the carbon cloth is where the reaction happens. That's where water is split into oxygen gas and protons and electrons. That anode chamber is separated from the cathode chamber using a Nafion membrane, which is a cation membrane. And then we have a stainless steel mesh cathode in that cathode chamber, which will catalyze the reduction of water to produce hydrogen gas as well as hydroxide anion. And so in this case, the ammonium will move from the anode to the cathode through the Nafion membrane. So this is showing anode fed operation. In the cathode fed operation, we reverse this and we feed the synthetic urine through the cathode where the stainless steel mesh cathode is, and then the sodium sulfate electrolyte through the anode. So the first and foremost thing that you see here is data on how much ammonium is removed from each chamber when we use anode or cathode fed operation. And we find that cathode fed operation almost always in a shorter experimental duration or shorter retention time lead to much better removal of ammonium. And we think that this is probably because of competition in the anode fed operation from other cations that might be present in the waste, in the synthetic urine. So in addition to ammonium, if you feed the urine through the anode chamber, you would have other cations that might move from the anode to the cathode and maintain electron neutrality. Again, the efficiency of the process relies on selectively only moving ammonium. In the cathode fed operation, we don't have a reliance on that. And that's why we don't have to worry about other cations that might be present in the urine. Next, if we look at how much chlorine is produced, we never detected any chlorine in cathode fed operation. It obviously makes sense. You shouldn't produce chlorine in the cathode chamber because you don't have the potential to produce chlorine in the cathode chamber. But in this case, we also don't ever detect any chlorine in the anode chamber either. Now, for the case where we feed the urine through the anode chamber, this is the traditional case of how EAS has been proposed. We see that over time we detect both free and total chlorine, and we see a very large difference between free and total chlorine, and this most likely suggests that a lot of ammonium gets associated with chlorine, and you end up maybe forming some chloramines. But the other important thing that we see is that a majority of the chlorine production seems to appear when the anode pH goes down significantly. Remember that in this setup, the anode pH goes down and the cathode pH goes up, and so the reduction in the anode pH is what basically spurs the production of chlorine further. And so we have confirmed now that if you feed the urine directly to the cathode of an electrochemical cell rather than the anode, you would avoid chlorine production while also achieving ammonia removal at a fairly high rate. What I want to show further is that one of the things that you would expect when you don't have competition from other cations is that the removal rate will scale with the amount of current that you pass between the electrodes. Remember that we are passing current between the anode and the cathode to really do these reactions. And so this data here shows that as we scale the current, we have a linear increase in the amount of ammonium that can be removed and recovered in the system. And that's exactly how the system should be. We have found that we don't really always have a linear relationship when we do anode-fed operation, again, because of competition from other cations. But when we get rid of that competition, we will have a linear relationship between the amount of ammonia that is removed and the amount of current that is passed through the system. So where does this all lead us to? So we are sort of now exploring different options. Of course, this cathode-fed operation of electrochemical ammonia stripping cells there are advantages, but there are also disadvantages. One of which is that the effluent urine from the cathode will be at a very high pH. And I'm not showing the data here in the interest of time, but we've figured that the performance, the linear relationship that I showed between the current density and the removal and recovery of ammonia is directly dependent on the amount of salt we put on the anode. And we want to get away from that reliance. Again, think about this, that you then, in these systems have to have a chemical that is not now an acid or a base, but a salt that we need to add. And to get away from this, we have a few different solutions that we have thought of. What I'm showing here on the right is a schematic of how we think that the electrochemical ammonia stripping process should be operated, which is where we would feed the urine through the cathode chamber of these electrochemical cells, rely on the base that is produced from water reduction to convert ammonium that is in the urine to ammonia that can be recovered. I'm not showing here how we would recover that, but we could recover that through stripping columns. We could recover that through gas permeable membranes. So I'm not showing how that does, but the key here is that we affect a pH change that converts all ammonium to ammonia that could be recovered. And then we have a high pH urine effluent that actually goes to the anode chamber. Now the question here is, if you had chloride in the urine, that chloride would then go to the anode chamber and form chlorine. So instead, we use a combination of cation and anion exchange membranes to separate the two electrodes so that we can concentrate chloride in that center chamber, so that what's entering the anode chamber is actually urine that doesn't have ammonia, and we've also removed majority of the chloride from that urine. And then because of the low pH that we would produce from the anode, we would neutralize the urine, and we have a treated urine that is the original pH that we started with, as well as having removed and recovered all the ammonium, as well as removed all the chloride that was in the urine. So we are currently performing testing on this idea. We've already shown that this works fairly well with other animal processing wastewaters that we work with in our lab, but now we are extending this to synthetic urine as well. With that, I would like to close my talk and see if there are any questions.
Wonderful, thank you for that. Give a moment for questions to come in. Here's one. At what, this is from Patel Asfaq. At what pH can we recover ammonia as a gas from urine?
Yeah, so you would have to go significantly above the pKa. I didn't show here the pH data, but what we've seen is that once you pass a pH of 10 is when you would have really highly efficient recovery of ammonium. The data that I showed on current density clearly allowed us, so the pH data from those current density experiments tell us that you get to a certain pH above which the rate rapidly increases in ammonia removal and recovery. So,
another question from Alex Milne is, are enough H2 or H clusters liberated to be of concern for explosions, etc., at lab scale, industrial, or closed environments like the International Space Station, for example?
That's a great question, Alex. So we, again, what I showed today was basically a narrow aspect of what we do in terms of urine, but the overall idea is to use this for things such as closed environments like at the ISS. And there are a few things that you can do with the hydrogen. So first and foremost, if you use a stripping column that is adjoining that electrochemical cell, you can selectively recover ammonia gas from the gaseous mixture. The effluent from the electrochemical cell will be a gas mixture of hydrogen and ammonia. But you can selectively remove ammonia from that using a stripping column, using selective gas permeable membranes. And then what you're left with is hydrogen. One of the things that has been proposed, this is something that a group in Netherlands, Bert Heimler's and Cees Buurtman's group has proposed, is that you can actually use that hydrogen as your anode reactant. So I mentioned here how you would have to apply a voltage to be able to pass electrons between the anode and the cathode. But if you keep recycling that hydrogen within the system, so hydrogen that is produced at the cathode goes to the anode, then you can actually oxidize that hydrogen. And the voltage of this system would be such then that in this case, you would, rather than adding voltage at what would be required to split water in water electrolysis, you would require a much lower voltage because you are now relying on hydrogen oxidation uh, to supply the electrons. And so there are strategies on how you would manage the hydrogen, especially for closed environments, like in space missions.
Great. One more question, then we'll go to questions for everybody. This one is from Utsav Shashvat. So the question is, do you see any precipitation in the cathode chamber due to high pH?
That's a great question as well. And so again, in the interest of time, I didn't show this data, but when we did those experiments with different current densities, as you go to higher current densities, the pH goes up higher, and over a longer term, again, I was showing data predominantly from batch 4-hour experiments in this case here. But as we have operated these systems for longer durations and at higher currents, we have noticed that you have precipitation and you have to work around that issue. But again, this is a matter of figuring, at what rate do you want to recover and remove ammonia, and maybe there is a sweet spot there where you can prevent precipitation while still recovering ammonia at a fairly high rate. One of the other things is that in this particular system that I showed, we had a set specific surface area of electrodes to volume that we were working with, and you can obviously look at that as a way of optimizing the system as well to avoid precipitation while also still achieving a certain rate of recovery of ammonia.